G'day, good evening, hello, wherever you sit and listen. This is a medical cannabis show dedicated to the legal practices and organic practices. So if you would like any illegal things, it's not the show for you. Or if you would like unsafe practices, it's not the show for you. I only teach organic practices from my studies through my study at university through plant soil science and microbiology, and I only promote safe practices of using medicinal cannabis and medicinal mushrooms as well. Thank you for the intro. And today it's an open topic. There's the screen, open topic again. See what pops up. There's a few people in chat, so I want to say hello. G'day to Vin. Hello to Jeff Papalia. Thank you to Sunshine Rainbows and g'day for the money. You put a little sponsor thing there. Thank you for that, mate. And while that's up, the little money sign underneath is there's 4 types of promotion things that you can click on afterwards. And that's for a thank you is for, I think, about $5 to $8. Next one's down to 80 for I'll give you my private lectures, private lists. There's about a dozen that I haven't released. And the next one's, I think it's 100. And that's for one video a month if someone needs some medical help and I can give them my opinions, unprofessional or professional opinions, it depends how it is. And the fourth one is for $300, and that's for a business or an organization, something to starting up. Or if you've got many questions you want to try and start up or go try and get legal help through medicinal practices, I can maybe give my input. So pass on to that if there's anybody wants it. Get back to chat. Yeah, Sunshine, thank you, mate. Very good. I can put that up now. Dave, 1969, how you going? Ned Kelly, g'day. It's an open topic, guys and girls. So we're looking for something to speak about. Otherwise, I'll be sharing what's his lecture this week? It was on soil. It was on the air gases, which is the temperature and the different gases that are involved in the substrates. So I can just share some slides of them. Hello, Supreme Grape. Cheers, everybody, says, or her says. LD6. Oh, this might be a question. What are the laws in Australia? Are there complications? I don't like speaking on what the laws are. Is ACT, it's sweet to have 4 plants. You can grow your 4 plants there, and pretty much everywhere else, good luck. South Australia has a little bit of leniency. Northern Territory used to have a couple of plants when I was living there you can have, and that's not there anymore. But everywhere else is like good luck. But we're nearly moving into a grey area, mate, because if you've got your medicinal license and if you have a few plants and get in trouble, I'm sure the judge would go lenient. I'm not by far means not a lawyer, but that's from my understanding what the laws are in Australia at the moment with the complications. So yeah, it's just best to try and look up and respect your laws. They do change. Wikipedia, I find a trust pretty good. And if need be, I suppose you could go into your prop shop just and honestly, what's the laws? Get not probably to your local one, but just go into one where you're passing after work or something and just politely ask the laws. For your friend that wants to know. Good question, mate. So it's fairly relaxed then. I wouldn't call it relaxed. Well, the law's the law. So if you don't respect it, there can be someone to enforce that law. And if you're not respecting the law, you'll get in trouble. So if it was me, I'd just respect the law, but I'm not telling people what to do. I'm just giving people options and saying safe practices and the right way to do it, I suppose. And that is to respect the law. But anyway, I don't like talking about the law. It was a lot better in Canada. When I lived in Canada for 5 years, it was just so easygoing, very easygoing there. But the law in Australia, it's very militant and very to the point. Yeah. All right, well, I'm going to start sharing some. Where's Sunshine? I really appreciate that. There you go, Sunshine. Good on you. Thanks, mate. I'm going to get and share some of my Yeah, it's not relaxing in handcuffs, the big, heavy, bloody things they are. That's right. Yes, Jeff. So it is best to try and respect the law, mate. That's my suggestion anyway. Do what you want, though. I'm going to share some soil science, I think, from this week, because the questions aren't really coming thick and fast. Share screen. One of the classes that I'm taking this week is called Soil Science and Technology. And this is week 3, I think, lectures. All right, the type of outdoor tillage. Tillage is sowing of the tops of the soil, breaking it up to prepare it for the preparation of soil for planting and cultivation of soil for after planting. I want to skip. Come on, soil and till health and soil health. Soil health depends on aggregate formation, the stability of it. Can it flow? Is it going to get washed away? Its density, is it compacted? The soil moisture amounts in it, is it retained so roots can breathe? Degree of aeration, same for roots of breathing and microbes. Rate of water infiltration, can it go in, or is it very clayey, or is it very sandy? Can it drain away fast? Is it too sandy and not enough clay? And the capillary water capacity, the amount of water it can retain. Conventional tillage is not really that good because it breaks up too much of the soil and leads to erosion through wind and through water and rain. But the go is conventional tillage, and that's where you do between 2 and 10% of your topsoil, and it wants to kill weeds better, it aerates the soil, and it incorporates a good soil organic matter ratio because you're putting everything that was on top back into it. It also controls the clod size so it doesn't get too big. That's the size of the soil particle. That's good for weed control. It incorporates, no, we don't want to use fertilizers. No, we use organic matters and organic practices. Puddling, it reduces. Leveling, of course, yeah, will help leveling. Yeah, puddling is not for medical cannabis. Soil conservation, soil tillage, very good. Conservation tillage, yes, just how it retains a lot of the stuff, and you've got a nice mulch layer already on top of the surface. No-till is another way, which is very good, where it's just plant. You don't till, you just go along and plant. See how it's just a bare place? Just keep planting. You can do that in medical cannabis by putting one there, one there, one there, then by season, one there, next season there, next season there, next season there. Soil crusting, that's not, you don't have that. If you've got anything looking like that, good luck. Problems in soil, crust layers, yeah. Management of soil crusting. There's more management. You don't want to really break it up like that in medical cannabis. Yeah, you want to mulch it. Just mulch it. And that will bring back the organic matter as well into it. Keep it as no-till as possible because underneath there is all the biology and fungus. And that's nice established layers. And now it's gone mixed up and it's got to re-establish itself. So you don't know what's going to move in. If there's a problem, if there's heaps of water, you might get pythium building up. You might get problematic things from doing it this way. So I suggest doing the organic way and mulching. Soil conditioners, yeah, you don't really want them. Oh, you do. I mean, you do want them in different ways, but you tend not to really use them if you can. Soil stabilizers, because you want a good organic matter already established in your soil, in your substrate. Soil density, particle density. Am I going into this too much? I might just stop sharing and see what's going on. Down to stream. I'll scroll up. Where are we up to, Jeff? Oh, really? It's got that on the screen? Pretty sure they're not going to take tax farms. Gobby Cannabis Vaporist. Hello, Mr. Vaporist or Mr. Vaporist. That's the go. Vaporing's, yes, I vaporize too. Very healthy. There's Teo, the American one. You're going over there in your stormy lands. But sure, if Australia could do that, we would. LD6. Well, here in the UK, they take all equipment and product, but if they can't find evidence for sale, they can't do anything. Wow, that's pretty good. They're starting to relax the laws in the UK a fair bit. Wish Australia would, mate. Jeff, Tyler, just reading, going down the chat. They're saying hello. How you going, mate? And Ned Kelly, how you going? All right, I'll get back to this. Some slides. No questions yet. Just present, share screen. Just need soil density. Yes, I'm not going to really go into this determination of soil density calculations to work it out. Yes, factors affecting soil bulk density. It's written dB, but it's bulk density. That's its maths terminology. Yes, clay in 2 sites. You've got to get your soil substrates correct. Really, this is saying it's 2 macropores, 2 clay, it'll stick together, no aeration. It's 2 micropores, it'll leach out and won't be good enough. So you've got to really fine tune it. Organic matter is the go. The effects of organic matter. Organic matter helps to form a good aggregation, so it reduces the bulk density, which is the compaction of the soil. Too compact, nothing's going to move, everything's going to come to a standstill, and it's not going to be very good at all. Depth of the soil profile, pore spaces reduces with the increase in soil depth, so bulk density generally increases as well. Medical cannabis only grows in the top sort of 0 to 60 centimetres, the first feet to 2 feet. If you're growing your trees for a seasonal thing, it might go down to 2 to 4 feet, which is 60 to 120 centimetre. You can see the soil profile here, where the tractor's probably grown over this section here, and in this section it hasn't. So it's compacted a hell of a lot in this section, so the bulk density is a lot higher. So it's not very good, limiting a lot of things. There you go. Nicely drawn in, very compact in there. Oh, here we go. Vehicle tyre pressure. See how the difference in if you've got a fat tyre pressure, you're going to spread the load, so it's going to reduce your bulk density. Otherwise, if it's skinnier, it'll push down more and really tighten it up. Not very good. Influence of bulk density on soil strength and root growth. Yeah, so the more higher bulk density, the more compacted it is, it's going to be reduce the root growth and soil strength because it can't move around and establish itself. Because fungi, bacteria, they all put out little bits of biofilm that aggregate up and attract soil components. The more clay present in the soil, the smaller the average pore, greater the average. Oh, sorry. I'm doing this for you guys. Porosity. This is soil porosity refers to the percent of soil volume occupied by pore spaces. So the ideal soil would have a total porosity of about 50% with equal amount of air to water filled. So total pore spaces includes both air and water. So that's why you've got to mix with clay balls or perlite or peat or something to really aerate it and give it its pore spaces, because that's where everything moves around in the pore spaces. So you want your biology to be established and not be in anaerobic conditions. No, this is a good example. So a brick has very, very high bulk density but low pore space, and a sponge has low bulk density but a high pore space. So you'll see how it forms like all these little microbe fungi homes. Not fungi homes, more so microbes, bacteria. Factors affecting total spore compaction. Look at the root growth. Struggling as, here it's struggling as, and here it's able to establish itself. Size of the pores. Depends on the 2 shapes. So you can go into classification of the pores if you want. I'm not going to read out all these stuff. It goes from your macropores, your mesopores, micropores, ultra micropores, and your cryptopores, which are so small you'd have to get it under the microscope. And generally, you want your micropores. They're found with within soil, and they retain water for plants to use and accommodate most bacteria. If you go up a little bit higher, for your mesopores, they retain water for drainage, which is really important. They also accommodate fungi. So that's why you want your difference. And your macropores, they're mostly the ones that you want, and water drains by gravity, effectivity transmits through the air, so the gases can be released, good exchange, large amount to accommodate plant roots. So that'll be probably on your topper layers, and it goes down. It'll go into these layers as your soil horizon develops in your pots and in your fields. Go through all this. Volume distribution of soil separates and pores in good soil structure. So you can see as the soil goes deeper, this is looking on the side cut, so you've got your organic matter, and it goes down and down and down. Cultivation of pore size. Yeah, you have to make it so you can add soil organic matter. So your organic compost, that type of thing, will help aerate it. Rupture resistance, wet consistency, stickiness. It's how to tell what sort of soil you got, how sticky it is. There's a test you can do just manually. Plasticity. Soil obtains different behavior characteristics when it starts to change its state as per the Attenberg limits on the screen. Plasticity, we're not into the soil testing. Soil water importance. Well, this is extremely important for soil physical, chemical, and biological processes. And remember, in soil, it's got the triangle. It has physical, chemical, and biological processes. That's the three things that make it up. And if you've got those three things right in the middle, that's where you've established a perfect structure. So weathering of materials and decomposition of organic matter in the soil. All right, let's get into it. I'll stop here and soil water. Anything else going on in the field? Hey, I see Tribro there. There you go, buddy. Where are we up to up here? Jeff, Tribro. Cheers, everyone. How you going, mate? It's nice to see you. Are there any cases where the soil is not dense enough? So what would happen? Yes, if the soil isn't dense enough, say if your bulk density is too high, everything's going to run off. I'll put on this one. Everything's going to run off. So the wind will erode it, the rain will erode it, so the slightest little adjustments will erode it. Is there any case where it's not dense enough? Yeah. You want the mixture. Like the beach, it's too sandy. It might be dense, but it's going to filter too many things. And the clay, it might be compacted where it's too dense and it's not going to allow water to percolate down, to infiltrate the subsurface. I hope that helped, mate. My soil's crawling, yep. You want biology to be established, very much crawling all over the shop. Hey, Magma Seeds. There you go, mate. Nice to see you. A good person I haven't spoken to in a while. Just like this fella here. Feed the hyphae, says Magma. Yes. Sunshine, getting there. Thanks, brother. Yep. Magma. Broken foot. Yeah, the hyphae are the growth that the fungus put out, and then the hyphae join together. They're little hair things, and then they attach together to form the mycelium that you see. That's generally white if it's healthy, brown if it's not. Broken foot. Yeah, Sunshine. Yeah, poor fella. Hope you get well, mate. Broken his blooming foot. Jesus. And a friend. Oh, that's not good. That's not good at all. I've been doing chop, chop, and drop, says Magma. Leaving things where they grow so they can adjust the soil where they grow. That's really, really healthy technique by doing that. The only problem is if you've got some sort of issue, if you've got a virus or fungal or insecticidal, some sort of infection or disease happening it'll reestablish itself. So you can only do that when your plants are in good, healthy conditions. But yes, generally that's the practice to do, for sure. You have healthy, and because you're doing a mulch layer with all the nutrients that are trapped in the leaves that they've got. Because remember, the dry matter of leaves still has a lot of nutrients retained in it. The DNA has heaps of phosphorus. There's other nutrients that are alive, and not much nitrogen left, because that's the greening bit. It's withered out of it, but there are other nutrients that are available, and the plants can be used once they're broken down. Amos, how you going, Amos? Nice to see you, Amos. Mostly wild weeds. Seems like different appears in conditions. Cool listening in. Oh, that's good. Thanks for the support. Magma. True, bro. True, bro. Aussie right on, brother. Yeah, true, bro. Woo. All right. Amos just got over a heat wave, 40 near all week. Yes, down the south of Australia, it's been putrid. Full sun. Had one bunch where they burnt baby bug sites. Oh, that's no good. Yes, you've got to keep, when the heat comes out, it gets very dry. So you've got to maintain the water so there's no burning of anything. Or if it's too full on, you have to, like you, when you sit outside, put up a blooming umbrella. I'll get back to sharing some soil stuff. 20 minutes. Maybe 20 minutes. Cheers. Soil water importance. Its importance is water, soil water. So you don't want it to leach out. You want it to retain it. You don't want surface runoff. You don't want, well, evaporative transpiration is going to happen. Air and water balances, rates, and yeah, it helps with soil temperature, helps with soil metabolism of soil organisms, and the capacity to store the water for plant growth. So the structure and properties of water, I went over this a few weeks ago when I did the big water show, so I'm not really going to go into it again. It's this hydrogen bond. It's so thick that it's just so hard to break. That's why it has a good bonding, cohesive, adhesive surface tension capabilities. Capillary action. Here's a test you can do in the at home. Science test. It's pretty fun. Just to prove capillary water, how it does suck up, and that's how water moves through, helps to move through the plant. Cohesive tension theory. This is also when you see around banks, it has an example of how it moves as well. So that's the dark patches around all the streams and the ponds outside. That's its cohesive tension theory, how water moves vertically. Soil water has an energy, kinetic energy. So when it's broken, it's changed its form. When soil goes from water, when it goes into vapor, there's a latent heat that's formed when it's formed into a gas. So it can heat up, and same when it goes to, there's a heat loss when it goes into its osing capabilities, when it changes form again. Forces acting upon soil water: capillary, absorptive, gravitational, and drag and shear. 3 models of water distribution. Yeah, good. Soil water potential. This is better explained. This sort of gets a bit technical with the matrix potential, the osmotic potential, and the gravitational potential. Actually, I'm going to skip it. It's really total water potential. Yeah, you add them all together, those 3 things I just said. And gravitational, hydrostatic potential, matrix potential. The attraction of water to the solid surface gives rise to the matrix potential. It's always negative because water attracted by the soil matrix has an energy state lower than that of pure water. Because you have a slight negative charge with pure water, and soil has a slight negative charge as well. So you've got that slight 2 charges there. When they can work against each other, or they can work with each other sometimes, they form different, like he's saying, different pressures. Expression of soil water potential. Water characteristic curve. So you can see how the volume, soil water potential down the bottom here. So it has a very low potential in sand, No, actually, no, I'm not going to explain that one. Hysteresis. That's when things want to, it's the relationship between soil and water content and matrix potential of a soil upon being dried and re-wetted. It's where it changes its state. So it goes from its drying to the wetting to field capacity to permanent wilting point. No. Yeah, field capacity to permanent wilting point. It's in the plants. Models. Oh, righto. Measuring water content. Water that may be evaporated from the soil. You heat it up. Example. No, neutrons scattering. This is how to measure it. Soil moisture. I wouldn't measure it using that. It's got radioactivity in it. Time domain reflectometer. Measuring soil water. I use a capacitive meter. These resistance blocks, they're cheap, but they need calibration a fair bit. And they come out with the Davis. It's a really good weather system station that I've used. So where's the capacitive resistance blocks? All right, maybe not going to show them. They're like a U-shaped one, and it's soil moisture meter, and it comes up on the screen like that. And it's a capacitive flow, so it doesn't work with resistance, it works with capacitance. So it's different electrical setup that's reliable, more reliable. Tensiometer, neutron. This is some methods of measuring soil water. So it just goes through their different ways that you can do it. And it's different ways that it works. I'm not going to go through one bit of it because I've explained the one you should use with your medical cannabis. We will cover numerical problems. I need some example. No, I'm not going to really, there's no point going through them. That's it. Go back to the screen. Hope there's some questions. Half an hour. Cruising for. G'day, wild weeds, listening in. Heaps. AussyCC, how you going? Just over 40, heaps of water. Any tips? LD6 says, any tips for indoors? Can't do anything outside here as it will either drown or freeze solid. You can, any tips for indoors? Create an environment like a baby wants to live. So if you put a crib in there or a baby basket, pretend it's a baby, and set your area up around it accordingly is the fastest way to say it. Would that baby whinge at the temperatures? Would it whinge at the humidity, the rain, the bugs, the smells, the gases? So that's the fastest and easiest way. I'd summarize that by saying, mate, please ask some more questions if you want to fine tune it. LD6. Magma says, rocks can also insulate and protect against heat and give nice extra heat off during the night. Yes, well said, because temperature has a lagging effect. Magma seeds, well said, mate. Temperature has a lagging effect where, just like in the seasons you'll see outdoors, once the season starts, it doesn't instantly get hot. It has a lagging effect, and that's why it carries on for extra time. The same with heating up things, like a brick wall will absorb it, and it will be cold at the start of the day. By the end of the day, it'll be way hotter than the outside air temperature. The same with rocks. So their size, and that's, we were working out those calculations before, it depends on the size of it to how fast it goes down. But that's exactly correct. So rocks can insulate. So if you're using, for instance, your Northern Hemisphere, using the Northern Hemisphere, so if you're a UK fella, you can put rocks outside and use them to attract the heat. They've also got camping showers that way, where they use black to attract the heat, because it goes on the reflectivity, which is your albedo. It's called the albedo of an object, which is your amount that it reflects. And like snow is very high, 90%, and flat white paint is 92%. So like mylar, I think it's 94% reflectivity. So the black absorbs it. It's the opposite end of the spectrum. So it, yeah, anyway, go on about that. Enough. Good words. Thanks, Magma. I'm using white quartz on the floor, Magma, in the greenhouse. Works a treat. Yeah, wicked. So the rocks are going to absorb it, and then the white is going to reflect it. So being in a greenhouse, You get long wave. Once the sun's rays transforms through an object, it turns into a long wave instead of a short wave radiation, and that allows the heat to build up faster. It turns into an infrared spectrum. So by him doing that, he's allowing it to keep it inside and bounce it around a lot better. Good on you, Jeff. Hey, Jeff, too, I've hit you up a Zoom invite about a week or so ago. It's, I don't want to say what my email is. Magma, your greenhouse looking fantastic, Jeff. Yes, I'm sure it sounds brilliant. Every day, how you going, mate? Nice to see you. What's the white quartz for? The white quartz is for on the floor to absorb the heat and to help with water infiltration. It's not going to get boggy. And the white quartz is for reflectivity. So it's higher on the albedo than darker objects. So it's going to bounce light around a lot better. Did you ever find anything, Magma says, like those red screens, Jeff? Red shade cloth? Yeah, they've got in the Newcastle University, they've developed this red shade cloth where it's very thin that you pull it over greenhouses, that it transforms the wavelength on a shady day into only usable energy wavelengths that the plant requires. So it's pretty snazzy. So if I was in a very shaded area like you, LD6 in England, you should be looking into that red shade cloth that Magma Seeds is suggesting. It's fantastic technology. It's so thin too. Excuse me. Okay, not sure what that's referring to. LD6, any help regarding room temperature, change cycles, soil types, any added nutrients you recommend, strains? Holy moly. Righto, I'm going to skim across this. So room temperature, pretend it's a baby again. Everything pretend it's a baby is the best way to think of it. If you had a baby in there, would it be whinging? So room temperature, you have to dial it in. So first get a few gauges to know what you're at. So get yourself a humidity temperature gauge, and then you'll know. If you can get 2 or 3 to stick at the high and low points, and then use a oil heater, being where you are. That doesn't emit any gases. If you've got kerosene heaters, for instance, they will emit a ethylene type signal, and that can finish plants early, as an example. So stick with oil heaters, and they're very economical and sustainable for the environment, which is the safe practices that I practice as well. Most of my studies is environmental science. So I like to keep those practices up and protect sustainability. Temperature change cycles. You can modulate that through your insulation. So you have to insulate the walls if need be. What did I do in Canada? I even went down and bought some of that thick poly stuff and put that around. So just get big sheets of that and fit it, just stick it next to your wall. That's going to insulate, like Eskies do, when you carry your drink coolers. Temperature change cycles. Well, that'll help that insulation. And mulch. Mulch helps your substrate temperatures too. Soil types. Use organic compost, 25%, organic worm castings, 25%, and perlite, 50%. That's your soil. Any added nutrients you'd recommend? No. You're just starting. If you're just starting, you might have to, mate, because it's really hard to get into organics just starting. You've got to go through and make many mistakes for the first 2 years with growing, and then you can branch out into organics, because it's really technical. You needed probably a lot more input or a lot of help. You can jag it, but you have to get everything right at the start, and it's really hard because you don't know what you're sort of dealing with. So I'd suggest even some bionutrients. I can't believe I'm suggesting this. Yeah. Anyway, Green Planet. I used to suggest Psycho, but they've been bought out by Montana now. Monsanto, sorry, Bayer. So don't use them. Only Green Planet. It's the only ones I suggest if you have to go. Any added nutrients, and only use that for the first 5 grows. After that, I'd like you to branch out into organics, mate, because you're supporting water. You're buying predominantly water with a few nutrients in it. What you want to do is You want your organic compost to be turned into microbes and fungi to be turned into plants' nutrients requirements. So the exudates will talk with microbes and fungi and release what it wants, and then that will prompt the microbes to do those practices to get hydrogen, because for them to get the exudates, which is high in organic acids, and the hydrogen protons, which is what the microbes want, they have to exchange something. So they'll exchange stuff like that. Strains you grow well indoors. Genetics is a massive, massive thing. So I'd get something off your friend or something along those lines. Genetics is just a big thing, mate. It's got to be, if you get genetics that's been grown, seeds have been made outdoors and you want to put it indoors, it might be hard for a first-time grower. If you get greenhouse seeds that be made and indoors, it's changes state because they get different UVs. Their UVB is blocked through greenhouses predominantly. So just get some good genetics that have worked for others indoors is the best way I suggest. Hope that helped out, mate. Please ask more questions if need. That was a pretty major summary. Wow. All right, back to reading up in the chat every day. Yeah, thank you. I've been hearing positive things about those red screens. Yes, because the benefit with that is that it's on shady days when the PPFD is so low that it's really not doing anything. These red screens will adjust the spectrum, pull it into one bright fine. It's like a magnifying glass. They'll fine tune it so it goes into the plant's, into its, 600 nanometer spectrum. It really fine tunes it into that array, benefiting plant growth. So if you've got it goes on sunshine, sunshine hours, daylight hours, sorry, in agriculture. So if you've got an area with low daylight hours, meaning that the sun hasn't come out and it's really been hidden a lot, you've got a high chance where this red screening will improve your yield and growth. The plants will love it. Could you use this all year? Well, it depends if you're, I think you're talking about the red screens. I'd be using it when I'm in the shady times because it might pull down. If you're in a middle of summer and you've got the red screen up, it might filter out a lot of the other spectrum that the plant uses. Because remember, it goes beyond 700 nanometers, the far red spectrum. It goes right up to 3,000. Remember that? Oh, yeah, I'll pull it up. I haven't got much to talk about. Nearly finished, so present. Where is it? Where are you? Spectrum chart. Plant science, I reckon. Yeah, I'll share. Say I'm not just thinking, who the hell is he bloody talking about? Share screen. Spectrum chart. Just share the whole thing. Stuff it. That one, no. Spectrum chart. Maybe it's under photosynthesis. I know it's a bar graph curve. That's all I'm looking for here. Photosynthesis. I'll try that. There it is, photosynthesis. All right, where are your charts? Is that it? There it is. So if it pulls it into this red spectrum here, you've got in your 600 nanometer, it's not, anyway, that's what it says. It's, was green supposed to be 500, I thought. Anyway, so you've got all this extra bit that the sun that it can use. So look at that, right up above the infrared, it goes above 700, it goes right up to 3,000, the thermal infrared, whereas so little is used, but it still is used. There's gases that are involved through that as well. So 52% is near infrared from 700 nanometers to 2,500 nanometers, the solar radiation curve. So 52%. So possibility you could be dropping that down. There's something here about the greens. Yeah, there you go. So that's a predominantly of plant photosynthesis, its spectrum range, but they haven't allowed for this long curve because a lot of meters don't test up for these long curves. What else we got in here about circadian rhythm? Yeah, that's cool. It's so true. Chlorophyll, yeah. It's where the chlorophyll gets made of different spectrums. This is some cool experiments. Examples of different light treatments on plant performances. Very, very cool. Anyway, I was not looking at that. I'll get back to the question. Could that be used all year? So I hope that helps out with the question, mate. Get back to me if it doesn't. Modern Queensland was distributing it. Oh, yeah. I don't know about that. Hey, Aussie Auto, how's it going? Can't beat Mac 1 for brekkie. That's the way. That's the way. Very medicinal, mate. You look into TC for seed sops. Look into tissue culture for seed sops. I don't know what that means. I'm sorry, Magma. One of my autos already showing sex at 3 weeks. Sweet. That's good. Usually it's about week 4, isn't it? So it's heaps early. So that means you've got some, what's it, heterosis, hybrid vigor happening. LD6, cheers, CC. Oh, right, all right, good. Yep, make that room looking like for your baby, mate. Here you go. Acro Science Redshift Shade Cloth. So it sounds like someone in the Northern Hemisphere has made it as well. And it's called ACERO Science Redshift Shade Cloth. The link, Magma. Moderator. Add as managing moderator. Add as standard moderator. They've got a new blooming thing in there. A managing moderator. Magma Seeds, you can put that link in if you want, mate. I've just made you a monitorizing thing. Oh, standard operation procedures. What was it for? You look into tissue culture for seed standard operation procedures. Okay. That's the go. All right. We got anything else to mention? Anything else anybody wants me to look up or anything to suggest? I'll give you a little bit. I don't know what else I can talk about for the bit. I think it's been going all right. Please, because there's a 30-second delay between the streaming and between you get it. So I have got to give you a little bit. You can look at this background here, all the trikes. That's what you want. There you go. Magma's just posted the link for the Redshift Shade Cloth. So I haven't seen it or anything like that, but I know Magma and trust Magma is a good bloke. And yeah, so if you're interested in it, that might be the link to look into and mention Magma. Magma Seeds sent you. Very good. Thank you for your help, Magma. Because yeah, it does help, because that's predominantly what the plant wants, is its photosynthesis so it can grow. So if it's being an autotroph, that's how it works. So the more better to the closer to the spectrum that it requires, the better growth pattern that you're going to receive from it. So if you can go and emulate that, even on cloudy days, they will love it. And they won't know the difference because you're just transforming that wavelength spectrum, the radiation spectrum, into a usable one for the plant. So it's perfect. So it's really good. So thank you. All right. Has anyone else got anything more to suggest? Oh, Walking Lightly. How you going, mate? Nice to... Your timing is impeccable, isn't it? Just asking last questions because I'm just about to finish. It's been a bit of a, I don't know, what can I put up today's one as? I was really just going through my own thing, wasn't I? Soil science, just porosity. Yeah, porosity, really. And spectrum, plant usable spectrum. Didn't touch, only just touched into it, but I suppose it's something that could be titled. Aussie Auto says, yeah, they show sex at 3 or 4 week, but rather 4. LOL. Yeah. Well, they call, I've had them for 4 to 6 weeks. They're just showing the sexes means that you don't have to go and flip them early or anything like that. The LD6, you'll be interested in this, that's the plants from just growing in veg state, they predominantly show their sexes, what they're going to end up like. So you don't have to get DNA tests and all these sort of male, female hormone, different tests to do it. You can just wait for week 4 to 6, and you'll look at the nodes, and you'll see the nodes. They'll either have little round balls there, which you'll shit your pants and go, that's not what I want, unless you're a breeder. And what you really want is to see the two little, I can bring them up, the 2 anthers. Where's that photo? I saw it before. This might help LD6. This one. No. So many buttons to press. My poor hands. I'm diseased. I got permanent disability in my hands. So for me, this one. So these things are the anthers that come out of the perigonal bract, it's called, or calyx, as normally known, and the pistils, as we cannabis people know them, but they're called the anthers. And if you see them, that's a good sign that you've got your female genetics in effect. If you see little balls happen and you don't see them around the nodes in your 4 to 6 week stage, that means that you've got a male plant that's going to eventuate into a male. So it's not going to really give you what you want. Because being trichome farmers, the calyxes see around the outside here, it puts out heaps of trichomes where the males form into seeds and wants to reproduce. So it forms not as many trichomes. Actually, to be honest, there's been a few tests being done to say that the male plants do produce a higher amount, but it's not a, because there's seeds there, they take up space. So with the female ones, it mightn't be as close and dense as trichome count, but there's more of them in total, which is going to add to the total effect, giving you a higher total effect. Yes, I hope that helps. All right, walking lightly, we're up to there. Is the auto in a small pot? They tend to show sex flip when root bound. Oh, yeah, good question. I haven't got much experience with autos. I know I've pulled a gene out of an auto, a ELF gene, early finishing gene, which is dominant, has a dominant effect in the autos, and I put that into my cultivars, which it's rad. So I can get some of them legit finishing day 50, full red hairs, no more white anthers at all. Awesome. He says, no, it's normal magma. Aussie Autos has good branches already, so she's big for 3 weeks. Yes, well done. Hey, Aussie, I've noticed, American ones, I've noticed big differences in the cotyledons on a batch of my seedlings. Can they be indicative of vigor or something else? Is there any research on them? Yes, it's called a COT1 gene, the cotyledons, and if you've got, they can show different expressions, and there's been suggestions that they're for starting out the plant. So if you've got big, massive ones, that might have been in an area that was hard, so it might have been bred in a greenhouse because of the UV, and it's looking for more UV. It could have been bred in a shady area or a time when there wasn't as much sunlight and it's looking for more. If you've got a cotyledon expression when you get 3 or 4 cotyledon leaves, I've had the 3 before, tricot it's called, you can, because normally in cannabis we have two, they're dicots. It's same thing. It's a plant looking for more starting ability. So it's trying to express itself more. So it might have been bred in some of those conditions that weren't as favorable. I hope that helps. Is there any research? I don't have any papers or on that. The bit that I've got, where would that be? Probably show you a picture of my trichots. I hope that helps, mate. Yeah, I reckon that'll help. I've noticed big differences in the cots on my batch. What differences have you noticed in your cot batch? So your ones with you're showing a dominant COT1 gene have given different expressions. So that might mean that that gene is working in favor with other genes to possibly produce positive effects and faster metabolisms. It might be something to enhance the vigor. So it sounds like you've got to keep them, mate. You might have a few positive genes. That might be a good mother plant to breed with, Teo. Aussie Autos says, think I'll pop some photos next week. So autos should be done same time as I flip. Perfect. That's what you want. That's about one of the really rad times for using autos. If you're first setting up like LD6 in England, you'll put, when you're first vegging, you get some auto seeds as well. So you're going to veg, and by the time your veg plants are ready to flip, after about 8 or 10 weeks, you will have a successful flower crop. So you don't have to stress out. So that's the perfect way to do it, was what Aussie Autos has done there. Lene, good morning. Good evening to you. Hey, Montana, how you going? Cheers to you. Every Day says, do you stay away from Fen seeds? Oh, AA, Aussie Autos, he's saying, I suppose. All right. Oh, sorry. Oh, okay. All right. Do you stay away from Fem seeds? No, I breed Fem seeds. Only breed Fem seeds. The reason being is because the males, you might lose. The cannabis plant has about 35,000 genes. So when you're going to normally breed it, it'll pull out the best genes, or not the best. You want it to pull out the best genes and then have a 50% blend of them. So your F1 generation will be a 50% blend of both of your parents. That's what it should be. And then you'll find dominance in slight genetic traits, like with humans. You'll see humans, their offspring looks pretty alike, but they've got dominant traits. Same with your plant's offsprings. But when you inbreed them, they tend to keep a lot of their traits themselves, and they'll delete DNA codes. So it might have 35,000 genes, and because some of the codes are they're not complete, it'll just delete them when you do a fem breed run, when you inbreed, or when you breed back with a parent only, one parent, keep breeding back, or then you breed back with the offspring only. That's what's going to happen when you just keep inbreeding it. But the bonus with that is it improves its homozygosity, meaning that it's less chances to herm out on you, to have other traits. It's very dominant in the genes that you want. And that's how land races are. They're very homozygotic in nature. So they tend to not herm out very much. That's why a lot of breeders are looking for those genetics. So for feminized seeds, I promote them and breed with them. And another reason is because, mate, how annoying. I promote the law practices, safe practices. So if you get sent, say if the law in Canada, it's 4 seeds, or here in the ACT, it's 4. So if you get four seeds and they're males, you can't do anything with them. So I don't like having that happen to me, and I didn't like that hearing about that from people. So I just do fem seeds. So that's my route. And you might lose, and talking about losing the genes, here, Frank's a breeder too. He knows probably about, he can back me up what I'm saying. And you, in that sense, you're A lot of the breeders now, they're not worrying about this, and they're just pumping out new lines, mixing it with this. Do a pheno hunt, find the best one, mix it with something else. Next one, mix it with. Because it takes generations to inbreed. Like it might take me, out of 2 years, I might be able to do 3 inbreeds, you know? So I'm up to a 3rd generation. So it might be really a lot more sturdier than at the 3rd generation and less chance of herming. So if you give it any character stresses, abiotic or biotic stresses, its chances of going into its herm state and trying to reproduce itself is limited because of inbreeding it and increasing its homozygosity. I hope that helps, mate. I think I'd be repeating. Oh, there's been a few words since then. Who was that even for? Every day. Yeah. Hope that helps. Every day. Let me know if it doesn't, please. I use FEMS. Aussie Autos says, yes, mate, I got autos. That's good. You got autos. LD6. Yep, that'll help you out. Like Aussie Auto says, it's really yeah, it's beneficial for first time starting out a grow. My jealousy pancakes works rad. Magma says, do you know if there are any nutrient uptake that can be measured on veg plants to indicate potential THC production or terp? They do this to find potatoes with longer storage. Oh yeah, for the starch potentials in the potatoes. Nutrient uptake. Well, do you know if there are any nutrient uptakes that can be measured in veg plants? Oh yes, I do, my friend. I've been sourcing this. Sunshine knows about this. This is something that I can't wait to put in my little selling thing. My little, what's those people have? Their little selling thing where they sell stuff that I don't sell yet. I'd like to start one because I'm not making any money out of this medical cannabis, and I should be. I'm still not, unfortunately. Anyway, how to do it is, do you know if there are any nutrient uptakes that can measure veg plants to indicate, oh, it's potential THC production? Oh, hang on, I was wrong. To measure potential THC production, no. So, well, kind of, actually. If you were to measure the amount of sulfate that's in the plants, because sulfate is very highly linked with its chemical pathways to THC. Jasmonic acid's also linked to it as well. So if you were to measure those chemicals, you can find out, is your plant producing those pathways? Are they ticked or not? So are the flashlights green or are they red to stop? So that's one way. I'm sourcing nutrient kits to test substrate and vegetable plant tissues, I suppose, and also bud tissue, but most of all vegetative plant tissues. So then I'll know what it's stored, what it has at that particular time, what it's toxic or deficient in. So that's what I was getting to. But yeah, not from what I've seen, mate, but that's just from what I know. That's a few things that you could possibly do. Yeah, good question. I'd like to go into that a bit more, actually. The time's getting on here. Hey, Lene, how's it going? Lene, big wave, it's the way. Frank says 100%. Hey, he says, FEMS are better if from IBL. Damn, I'm not up with these blooming terminologies. I don't know, in vitro. I'm not sure of that. So Frank's a cool breeder from North America, and he's into his FEMS as well, so that's cool. Every day, very interested. So you can breed FEM traits and no sus stuff going on to make them female. That's pretty cool. Do you ever get males though? No. If you grab the pollen and then manual pollinate, that pollen should be feminized. So yeah, if your plant herms out and you get a little bit of pollen, you can stick that on a bud, and the seeds that you get should be feminized. So you ever get males? No. From all the ones that I've done, that I've bred, and I've probably tested at least 100, and I far out other people have tested, wow, probably at least 500 for me. I've never heard of a male come back. We can go into another thing. I love genetics. One of the shows I've actually put off is my next show. It's on genetics. I've got all these papers and studies. Actually, while we're talking about it, let's just, I'm going to show you. Where is it? Plant stuff live. Lives. Christy Holidays. Yep, right. Yeah. I'll just show you because I get excited with genetics. Because it's the future. If you know what's going on with genetics, you can manipulate things. So it's really rad. So I'm going to show you just my file. This is something that I'm going to do in about a month's time when I'm a bit more not busy. I'm extremely, extremely busy at the moment. So I haven't got spare time. Week 23. This is all my genetic stuff. So I was going to run through all this rad stuff with you. Anti-cannabinoid genes, and go into how it helps and all its different pathways, CBD to produce ECS, the endocannabinoid system, what it does for good medicine. Yes. This is it here. Yeah, I was thinking, hang on, I've done any cancer. I've done that. It's this one here, week 24. This is one I haven't done. So it's breeding and genomics, the genomes, fast breeding tools, disjointed policies in Australia, hang drying compared to not hang drying, plant densities, heterosis, which is hybrid vigor, how to maintain it and obtain it, human chromosomes under a microscope. Right, that's to do with ploidy, really, polyploids, because you can look at your plant's chromosome count under the microscope. It's pretty cool. Time for drought. Anyway, this is all this good stuff, like stuff like this. How to always get things like this. That's what I am looking for. Heterosis, or hybrid vigor, as they say. Like this chart says. Heterosis, or hybrid vigor, is a phenotypic trait, phenotypic result of gene interaction due to the effect of heterozygotics or of hybrids. So you can, while you get your heterosis, like, so that's what Aussie Autos would have got from his plant showing sex at week 3. The more homozygotic, here we go, the more homozygotic alleles, allele is meaning a gene, the more homozygotic genes are complementary in parental form, the greater effect of heterosis in the hybrid forms. Hence, the greater theoretic heterosis can be expected when there is a large allele diversity in the genes. So that means that from you've more chances of getting your hybrid vigor or your heterosis in non-homozygotic plants. But if you breed with marker-assisted breeding, which I do, you will select the vigorous ones, so you'll breed with those ones, maintaining heterosis in your homozygotic line. That was well explained. Right. That's just a chart. Anyway, this is all, I just love all this stuff because it's what we're about. It's how to improve things. Here's a, what's this? Normalized differentiation index on vegetative plants. So this might be similar to what you're talking about, magma, but this is to do with photosynthesis and the chlorophyll adaptation. So he was wanting to ask before how to measure the pathways through the jasmonic acid or the sulfate pathways, so to improve your trichomes production and your terpene production. So this is just a study. Anyway, I'm going on about it. Back to it. So yes, it's pretty cool. Hey, Teo, rad, I did help. That's good. I like hearing that. Kemet, how are you? Kemet 432. Magma, I think it would be a great way to run larger numbers and find out if they're flowering. Without flowering, yes. as he says underneath, without flowering. Sea green is the easiest way to phenotype plants that don't get too big. Yes. Wait, what? You can use herm pollen to pollinate a male on your next generation will be feminized. No, if you pollinate a male, you have to pollinate a female. So if you get herm pollen, that means it's showing the anthers, and that means you got pollen produced out of it, which is your little female bits to your, what are those little cells called? Pollen grains. And that's going to start a reproduction and look for an ovary to reproduce in. So if you get them from your herms and pull them onto your buds, your female buds, that's when you'll make female seeds. If you put your female pollen onto your males, that's when you'll get a normal F1 generation, not an S1, a selfing generation. You'll get a normal F1, which is back-crossed with one of the parents, which is a good way to bring out traits that you want. That's how you'll bring out, if you like your stinkiness, you'll choose the stinkiest plant and back-cross it with a parent. Breed those seeds. Choose the stinkiest again, back-cross it with that parent, and then keep it going. That's how you do. Oh, good, it helped. Sweet. Frank says, herms are female, and I tried explaining that the chromosome evolution happened before cannabis was cannabis. So female seeds will be not turn male. Yes, chromosomes are chromosomes. Kermit, I'm just skimming down here. Aussie Autos popping the primo. Self-pollinate. Aussie Autos will do. Frank says, hermaphrodite traits can be stabilized. Prime example of the hemp made for seed production, but they have been stabilized to self-pollinate. There you go. Anything, plants can be manipulated and worked with. That's why I've been playing with ploidy for the last few years for some tetraploids and some triploids, and to see what their expressions are and how I can manipulate those chromosomes in them to try and get some better outcomes. Once it's legal, yeah. Good on you, Aussie Autos. Respecting the law, mate. That's what we do, and I like to promote respecting the law. Sometimes people accidentally breed with hermaphrodites, thinking that they are male. They are feminized when accidentally breeding with hermaphrodites. Yes, that's right. Solar went up yesterday. Okay, very good. Every day, last one, legend brother, I'll be doing the same. How it is all bro. Okay, exactly, Frank. Hermaphrodites are stressed females. Yes, that's correct, because they're showing their traits. Their heterozygosity is high on the index. The little bit of stress you give to them, bang, shows their reproductive capabilities. Because whenever a plant's they're going into stress, they want to reproduce. So that means they'll keep their genetics alive. That's their tendency. An hour 10, righto. Thanks, everybody. I'll be doing the same this time next week in 7 days. Please bring your questions. It'll be an open topic again. I appreciate everybody for coming in and asking questions and hanging out. Good on yous. Appreciate it again. So happy breeding, happy growing, and not safe travels. Happy breeding, happy growing, and good health to you all. Yes, thank you. Good night, goodbye, good day to you.